Introduction. Welcome to today's lecture on the fascinating world of RNA. In this comprehensive course, we will delve into various aspects of RNA biology, exploring its diverse functions and mechanisms. RNA, short for ribonucleic acid, is a versatile molecule essential for gene expression, regulation, and cellular processes. Join me as we explore the intricacies of RNA folding, alternative splicing, non-coding RNAs, and much more. Let's begin our journey into the fascinating world of RNA. RNA molecule presents a remarkable capacity to fold into complex tertiary structures and secondary structures. Secondary structures, such as stem loops, hairpins, and bulges, form when complementary regions of RNA interact with each other through base pairing. These structures play crucial roles in RNA function, including gene expression regulation and protein binding. Understanding RNA folding is essential in deciphering RNA structure-function relationships and developing therapeutic strategies. Alternative splicing is a critical mechanism where a single gene can generate multiple RNA isoforms. This process allows for the production of distinct protein variants from a single genomic locus. Through alternative splicing, different exons can be selectively included or excluded, leading to the creation of functionally diverse proteins. The regulation of alternative splicing is tightly controlled and involves complex protein-RNA interactions and spliceosome assembly. Non-coding RNAs, RNAs, represent a large and diverse group of RNA molecules that do not code for proteins. These RNAs have gained immense recognition for their involvement in crucial cellular processes, including gene regulation, chromatin remodeling, and cellular homeostasis. Examples of RNAs include microRNAs, long non-coding RNAs, and circular RNAs. Elucidating the functions and mechanisms of RNAs is key to understanding the complexity of cellular processes. Ribozymes are RNA molecules capable of catalyzing biochemical reactions. This remarkable feature challenges the classical notion that only proteins are capable of enzymatic activity. Ribozymes have been found to perform diverse functions, including self-splicing introns, peptide bond formation in the ribosome, and RNA cleavage. Understanding ribozyme structure and function has broad implications in both basic research and potential therapeutic applications. RNA editing is a post-transcriptional modification process that alters the genetic information encoded within RNA molecules. Through RNA editing, specific nucleotides can be modified, inserted, or deleted, leading to alterations in the RNA sequence and, consequently, protein diversity. This process has been found to be essential in various organisms, playing critical roles in development, adaptation to environmental changes, and disease progression. RNA interference, RNA, is a highly conserved mechanism that regulates gene expression by specifically degrading targeted RNA molecules. Central to this process are small non-coding RNAs, such as small interfering RNAs, siRNAs, and microRNAs, miRNAs, which guide the RNA-induced silencing complex, RISC, to complementary RNA sequences. RNA has significant implications in controlling viral infections, gene therapy, and the regulation of endogenous gene expression. RNA modifications refer to the chemical alterations of RNA nucleotides, distinct from the primary sequence. These modifications include methylation, pseudouracilation, and acetylation, among others. These modifications can affect RNA stability, structure, localization, and translation efficiency. Investigating RNA modifications contributes to understanding RNA dynamics, cellular responses, and disease mechanisms. Spliceosome assembly is a complex process involving the interaction of small nuclear ribonucleoproteins, snRNPs, with pre-mRNA molecules. The spliceosome removes introns and splices together exons, contributing to alternative splicing and mRNA maturation. The assembly and regulation of the spliceosome are tightly regulated, with multiple factors involved in ensuring accurate and efficient splicing. RNA-protein interactions are fundamental for various RNA-related processes, such as RNA stability, transport, and translation. Proteins, including RNA-binding proteins, RBPs, play central roles in recognizing specific RNA sequences or structures and modulating RNA function. Understanding RNA-protein interactions is crucial for unraveling the intricate mechanisms underlying cellular processes and developing targeted therapies. RNA localization refers to the specific targeting of RNA molecules to particular subcellular compartments. This process allows for localized protein synthesis, spatial regulation of gene expression, and cellular response to stimuli. RNA localization mechanisms are diverse and can involve RNA binding proteins, as well as cis-acting elements within the RNA molecules themselves. Investigating RNA localization is essential for understanding cellular organization, development, and disease. Retroviruses are RNA viruses capable of reverse transcription, a process where viral RNA is reverse transcribed into DNA. This DNA is then integrated into the host genome, leading to persistent infections. Understanding the mechanisms of reverse transcription and viral integration is crucial for developing antiviral strategies and combating retroviral diseases, such as HIV/AIDS. The RNA world hypothesis proposes that early life on Earth could have relied on RNA molecules for both genetic information storage and catalytic functions. RNA possesses dual capabilities, serving as both genetic material and enzymes, ribozymes. Exploring the RNA world hypothesis provides insights into the origins of life and the emergence of self-replicating systems. In this captivating tour of RNA, we explored its folding, alternative splicing, non-coding RNAs, and more. As researchers, understanding these intricacies expands our molecular biology knowledge and opens new therapeutic opportunities.